There are three, when we're talking about nucleic acids. Oh, sorry. Is it on there now? When we're talking about nucleic acids, there are two types of nucleic acids, but there are three parts to every nucleic acid, that two of which are always the same. There's a phosphate group. Phosphate, again, is a functional group. And it looks like this, usually a negative on each one of those. And of course, this oxygen binds to something. Remember, each oxygen forms two bonds. Oxygen forms two bonds. That's why this oxygen has two bonds. That's why this oxygen has two bonds, one here, one here. Phosphate can make 5 bonds, as you can see. But each oxygen needs 2 bonds. So this has 2 bonds, this has 2 bonds. This one only has 1. Has an extra electron, so has a full octet, so that makes it negative. So phosphate is negative. In fact, DNA is negative as a whole unit, has a negative charge to it. All right, so it is somewhat negative. Phosphate, this bond here goes to a sugar. This goes to a sugar. Normally, what we do is, instead of drawing all this out, everybody, I hope, agrees that this is really a big pain to draw all these little things. Normally, what we do is just draw a circle. So phosphate is usually, it looks something like this. Sometimes it's yellow, sometimes they do it in red, whatever. But they'll do just a circle, a big old circle, to let you know that they're talking about 5 atoms. 4 oxygens, 1 phosphate, negatively charged, double bond oxygen. All right. So you have a phosphate. That oxygen bond is going to go off to a carbon. So there's a carbon there. That carbon is the 5th carbon, the 5 prime. We call it 5 prime. Anytime you have a ring structure of carbon, we number the carbons clockwise. In biochemistry, in organic chemistry, it's all the same. When you have any kind of ring, so there's oxygen, that's how carbohydrates close the ring. The oxygen has 2 bonds, it forms 2 bonds. And it closes a ring structure like this. In the case of ribose, this is ribose, the general sugar ribose, it makes a 5-sided ring. It has 5 carbons in total. Has 5 carbons in total. And I'll count them in a minute. And then that 5th carbon is connected to an oxygen of the phosphate group. So that's where the 5th carbon is. So the 5th carbon, 5 prime carbon, is connected to a phosphate, always. We number them clockwise. So if you think of a clock, and I was just talking about this with some upperclassmen, people don't realize how to read clocks, but when clocks turn, they turn in this direction. I hope you realize that. If you don't, I hope you realize it now. They turn towards the right. We call that clockwise. So going clockwise, this is the 1 prime carbon. The second one, obviously, is 2 prime, 3 prime. I like to think of this as 0, right? Because it's an oxygen. It looks like a 0, so I think of it as 0. And the 4th carbon's here, 4 prime, and this is the 5th prime carbon. Now, at these positions, at each of these positions, you don't see it here, you don't see them, but there's an OH and an H at each of these positions. And they alternate up and down. So this is up here connected to this. I'm going to draw it, you know what, I'm going to draw it off to the side so we don't. So if we were looking at ribose, it would look something like this. It looked like a home plate in a baseball field, a pentagon, and then this carbon, which is here, this carbon, would be connected to a phosphate here. Well, each of these is going to have an OH and an H alternating. So OH connected to it and an H down. Now you got to remember, this is something everybody should know. Each carbon, which is represented by a dot, a center, every connection between each line, there is a carbon. There's a carbon here, there's a carbon here, there's a carbon here, there's a carbon here, and there's a carbon there. And at every joint of the circle, there's a carbon. It's assumed. We're not going to draw it. And wherever you don't see anything, it's assumed you understand that there's an OH and an H. Because remember, every carbohydrate has CHO. 1 to 2 to 1. For every carbon, there's 2 hydrogen and 1 oxygen. For every carbon, there's 2 hydrogens, 1, 2, and 1 oxygen. Every carbon here has 2 oxygens and 2 hydrogens and 1 oxygen.
Why do we draw?
Because it makes it easier not to. It's a shorthand. Like, why do you guys text with LOL? Why don't you just write laugh out loud? Because it's shorter. It's easier. That's why we do it. Yeah.
What is the relationship always be on the outside and the base be on the inside? Or does it really matter?
It's up and down. It's not in or out. So yeah, it does matter. It impacts orientation. It gets involved. You have to get. It gets in. There's a lot to biochemistry and organic chemistry, but no, for the sake of this class, it's not. It's not relevant.
Both of them have always been outside of the molecule?
They're up or down. So there's no inside or outside. There's no like wall. It's not inside. It's just one sticking up and one. You'll see tomorrow when we build it. So one sticking up. So you have a ring. So there's a ring, right? So here's a ring. One of these goes up and one goes down. Does that make sense? It's not really about in or out of the ring. It's either up or down. It has to do with organic chemistry. And so there's this rule, right, that electrons repel each other. So these atoms are all trying to get as far away from each other as they can. But they're kind of being pulled together. They're the opposite forces, the covalent bonds that are pulling them together. So they orient themselves to be as far away from each other as they can and still be together. That's how these things are. That's how atoms and electrons and why the molecular shapes take on the shapes they do. The basic force is the repulsion of electrons and the attraction from the sharing of bonds. So you have up and down. In this case, this will go down, I think, and the other goes up. It looks like it's in, but it's just up, if that makes sense. And then the next one, if I remember correctly, I think this one's also down, and then this one's up. And here you have hydrogen, and you might ask, well, where's its oxygen? There's a carbon, there's a hydrogen, but where's its other? You tell me where its oxygen is. Every one of these has an oxygen. Where's its oxygen? This one, yeah, this one's oxygen. So that's what makes a circle. And because it makes a circle, it doesn't have that second hydrogen's gone. So that's why it lost it. Then up here you have this carbon that's sticking up is one bond to the phosphate, the other bond goes to an OH. I think the OH is either up or down, I can't remember, but I'll just draw it. OH, and then the fourth is to a hydrogen. So again, every one of these carbons has 1 oxygen attached to it and 2 hydrogens, 1 to 2 to 1. That's every carbohydrate has that ratio. Yeah. Where? Over here. On this carbon. Yeah, you're right. I didn't draw it, but you're right. This you have another hydrogen. So H2O, right? Every one of these has 1 oxygen and 2 hydrogens. So that's the combination for all carbohydrates. So if you look at any carbohydrate, now will there be changes? Will some chemical reactions add another element to it, therefore get rid of a hydrogen or oxygen? Yes, but in general, it's 1 to 2 to 1. Everybody got that? All good. All right. So this is what it looks like, and so you have this 3'. So there's 2 other points. The 5' is obviously very important, but to the 3', you have sticking out. You have a hydroxyl group. Now, as you just saw, every one of these has a hydroxyl group for the most part, but this hydroxyl group becomes very important. So there's 2 really important places in a ribose. It's this 3 carbon, and let me use green. So it's a 3 carbon and not the 4. And the 5 carbon. So those are the 2 important junctures. There's obviously a lot. There's also 1. The 1's position is very important as well. But those 3 spots are very important. And then this one becomes kind of important. And we'll look at in a minute why it's important. But this is the 3' end. This is the 5' end. Becomes very important. 5' always has a phosphate. 3' always has hydroxide in DNA and RNA. The 1' is where we're going to attach the nitrogenous base. So the A, C, T, or G gets attached here. All right. Does anybody remember what's important about this, the 2'? Yeah. Very good, very good, excellent. So this is where we determine, is this deoxyribose? Oh, not bore, but bose. By the way, I want to remind you, ose means sugar. So, in case you're wondering. Or is it ribose? Is it one or the other? Deoxyribose means there's no OH there. It got changed, and instead of having the OH, remember I just said it's 1 to 2 to 1, with some exceptions, and deoxyribose is one of those exceptions. It's missing an OH group, so it only has hydrogen. So in the case of deoxyribose, the second position only has a hydrogen. In the case of ribose, that second position has the OH. So in RNA is ribonucleic acid. It has the same sugar, the same 5-sided sugar, the same connection to the phosphate. That's all the same. It has an OH group sticking out here. That's still the same. The difference is that in ribose there's an OH here. So again, this is different. These two are the same, but that is different because in deoxyribose, in DNA, you have the ribose sugar again, still ribose, still 5-sided, still 5-carbon, still connected to phosphate, just like RNA. Still has the 3' OH, just like RNA. But the difference is that this is just H and H, no OH. So that second carbon is different between DNA and RNA. That's one of the big differences between it. And believe it or not, that one difference changes things. Right? I know it seems small, but like when we looked at the enantiomers, the things are small changes in these molecules make huge changes in their function. So that's really the big difference there with the center. So when we connect them, what happens is that you get, I'll try to do this in color-coded method. So you have phosphate. So you have this chain, just like you're making at home, just like we'll put together here in the classroom tomorrow on Friday. You're going to have a chain that connects phosphate to sugar, to hydroxide. The hydroxide gets connected to what? To the phosphate. So phosphate, sugar, phosphate. What comes next, you think? Another sugar. And it just keeps repeating itself. What do we call units that repeat themselves over and over again? Close. A polysaccharide is an example of that, yes. But what we call something that repeats, it's in general, when you have a unit that repeats itself over and over again. Again, polysaccharides are an example of that. Say that again? Polymers, that's right. These are all polymers, and DNA is a polymer. It's a polymer of nucleotides. It's not a polymer of saccharides, right? And I can see why you would think it's a polysaccharide because it's a bunch of sugars. That makes sense. But these aren't considered sugars. These are called nucleotides. The monomer here are nucleotides. Nucleotides are what? They are phosphate, sugar, and nitrogenous base. I'll spell it in just a minute. There's sugar. The 3 parts to a nucleotide. I'll tell you in a minute. So just leave some space, and we'll come back to it. So there's this one this one space, there's one more bond that's available that your body and all life on Earth takes advantage of, is this idea of nitrogenous bases. Nitrogenous bases are, of course, bases. They have nitrogen in them. I'm not going to draw them out. I don't think it's useful. I don't think it's going to be on the test. Maybe I'm wrong. Maybe your test, all of a sudden, they decide to put the different bases on there. But I think what's important to realize here is that some of these bases are double rings and some are single rings. Remember that when we talk about rings, we're talking about carbons bonded to each other, forming these rings. So a nucleotide, we could define a nucleotide as being, let me choose a different color. We can define a nucleotide as being 3 parts. And I'll write it real small here. 3 parts. 1, a phosphate. 2, the ribose or deoxyribose. And 3, the nitrogenous base. So phosphate, sometime a deoxy or ribose sugar, depending on whether you're doing DNA or RNA, and 3, nitrogenous base. And then it's all in the name. A nitrogenous base has nitrogen in it. By the way, let's talk about the elements that are in DNA. What elements do we have in DNA? Who can tell me really quickly? What are the elements involved in this, now that I've told you all this? Carbon for sure is in all of them. Carbon, oxygen, hydrogen. So far, I don't see a big difference between this and sugar, CHO. What? Nitrogen. Oh, that's different. So what elements have carbon, hydrogen, nitrogen, and by the way, I've seen many AP questions based on this. So you really need to know what elements are in every biomolecule. So you have carbon, oxygen, hydrogen, nitrogen. That sounds a lot like a protein so far. Proteins have nitrogen, carbon, oxygen, and hydrogen. Phosphate. That's the big difference right there. This is the baby that tells you when you got phosphate, let me do another color. When you have phosphate and nitrogen, yeah, it's definitely nucleic acid. There are elements that have, well, not definitely, but likely, right? You should be able to tell, but there are other one thing you don't see. What is some of the things that are always missing? That's right. Never do you see sulfur in DNA. They are in proteins, but never in DNA. So make sure you realize that. That's kind of an important little point that will crop up and bite you in the butt if you don't know it. So 1, 2, and 3, these are the things that this is what we call a nucleotide. It's actually a nucleus. These two together are a nucleoside, but with the phosphate, it's called a nucleotide. I don't know if that's going to be important, but if you just had just the ribose and the nitrogenous base, we'd call it a nucleoside. If they had the phosphate, we'd call it a nucleotide. So, again, I don't think is it that important? I don't think so, but we'll see. So, again, that we already see the difference between the two sugars. The nitrogenous bases, the differences are pretty clear, but I'm not going to get into it. Again, I don't think they're going to ask you to tell the difference between C, T, and U. I do think that you should know that C, what's wrong with this thing? Is it frozen? C, wow, okay. Wow, all right. C, no?
Oh, so it's letting me move, it's just not moving.
Okay. It's freaking out right now. What's going on with you?
it hot?
Huh? Start? All right. So, no, really? No. What's going on? Oh, my pencil's dead. That's why.
Oh, you gotta charge your pencil?
Yeah. Yeah, it's a 0. Okay, that makes sense. I was like, what's going on? All right, so I can write with my finger. It's not going to be as neat. Too bad. All right, so when we talk about C, T, and U, these are single rings. So you're going to see, you can see the single ring right here, right? There's single rings. So that's either C, T, or U. Whether U, obviously, I hope it's obvious to you, is only in RNA. U is only in RNA. Where T is only in DNA. So you either have T or you have U. You can't have both. So in RNA you have U, and those are single rings. That's really key.
I remember this from that's what I was gonna say, PBS. No, I remember.
You better remember from honors biology.
Yeah, what they
PBS.
stand for?
You coming at me with PBS? I don't want to hear about PBS. I taught it to you in honors biology.
T is stand for what and U stand for what?
T is thymine, U is uracil,
Okay.
right? And C is cytosine. So these are the 3 single ring nucleotides. The nitrogenous bases define a nucleotide called cytosine, thymine, and uracil. Uracil is only found in RNA, and thymine is only found in DNA. U replaces T in RNA. So wherever you would normally find a T in DNA, you'll find a U, and vice versa, right? So that's key. These single ring things are known as pyrimidines. You may see that in your reading. So do I think they're going to ask you, is this a pyrimidine or is that a purine? I don't think so, but just know that they're pyrimidines. And just in case it comes in handy, it might come in handy. Their pyrimidines are single ringed. The double ring structures, as you can see here, there are double ring ones. They're known as purines. I don't know of an easy way to remember the difference between purines and pyrimidines. I just memorized that there's A is a double ring structure and G is a double ring structure. The key is that a purine matches with a pyrimidine when it comes to hydrogen bonding. A pyrimidine never binds hydrogen bonds with a pyrimidine, and a purine never hydrogen bonds with a purine. These two don't match. These two don't. None of these match. But these can match to these. So they match with each other. Pyrimidines and purines go together. That's the only way they're going to hydrogen bond. Does that make sense so far? No big issues with this?
I'm not confused.
Please, you don't have to explain yourself. Any question, please.
What do A and G stand for?
It's a good question. A is adenine. And G is guanine. Again, I'm sorry, my pencil's dead, so I have to wait till it charges. All right, are we good? Do you need to know guanine, adenine, cytosine, thymine, uracil? Honestly, if I were a betting man, and I'm not, but if I were gonna bet, I would say you're not gonna be asked to tell the difference between purine and pyrimidine. But you should know that there's double rings go with single rings. If you know that pyrimidines are single ring and purines are double rings, I think that might come in handy, maybe. Yes?
So, does this have to do with the fact that C and G are double bonds?
Yes.
So that's
The reason that C and G do double bonds, C and G do double bonds and A and T do single bonds is because of how many available hydrogen bonding spots they have. So if we drew it out and we can look at it and we could draw it out and we can compare, and I think when you look at C and G here, once you draw them in your model, and when we look at them in the 3D model, I think you'll see this more clearly. But when you look at C and G, there's 3 available spots for hydrogen bonding. So when they bond, they can form 3 hydrogen bonds. A and T only have 2 available spots, so they can only form 2 hydrogen bonds. Now, how does that make a difference? Well, that helped us understand Chargaff's rules. Chargaff discovered this, and I'll explain that to you in just a second. But Chargaff came up with the rules of this kind of bonding by measuring how much energy it took to separate them. And obviously 3 hydrogen bonds, it takes more to separate them than with 2 hydrogen bonds. Does that make sense? Does that make sense?
So, ain't it like I forget what it was, but it was like A go with G or something like that.
I just said, yeah, for sure. A goes with T and G with C. It's the base pairing rules. And the way we discovered the base pairing rules is because of how much energy it took to separate A and G versus A and T. Because they have a difference in how well they stick to each other, we were able to decipher that A always matches with G and A always matches with T and G with C. That's the base pairing rules. That's something that should be always in your mind without any question.
And so when it's RNA, so A pair with U.
That's right. A goes with U in RNA and G still goes with C. That's right. All right, so that's great for the general structure or the kind of background you're going to need. I wonder if this is paired up enough to work at least to the end of this class. All right.
You have to be done in 2 minutes.
Oh, that's all I need. So when we make DNA, we make a series of sugar phosphate bonds. It looks like this. It starts with a 5' on this end and a 3' on that end. It has the nucleotides, so you have your ribose sugar. One person has not left their phone during most of this conversation. I'm wondering what they're doing. So the sugar phosphate backbone, this is called the sugar phosphate backbone. Just like we drew here, right? Sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, all the way down. That's what we call the sugar phosphate backbone. So if you see that, you need to think about the fact that this is one long polymer alternating sugar. This actually should be phosphate, sugar, phosphate, sugar, phosphate, etc. Right? All the way down from one end to the other, 5' to 3'. That's one strand of DNA. But DNA is not a helix. DNA is a what? A double helix. That's because there's 2 molecules in every DNA. Every DNA molecule has 2 strands. I don't like that language because the reality is you have 2 molecules of DNA. That's the actual thing that's going on here. And there's a misnomer. We define it differently. We say a molecule of DNA is actually 2 strands because that's how we find it in nature. But if you want to get technical, these are 2 different molecules that are hanging out with each other. All right, so each strand is its own molecule. Because a molecule is defined by covalently bonded structure. This is a covalently bonded structure. Everything in here is covalently bonded. And remember, sticking out of it are these nitrogenous bases. You can have an A, you can have a T, you can have a G, and there's a spacing. That's why it's important you measure when you do this project at home. You measure because there's a specific spacing that's required for every turn. And you can have a C, give me a second. You can have a C, etc. On the opposite strand, the other strand, the way they have to line up geometrically in order to form hydrogen bonds, otherwise they won't form hydrogen bonds and they won't stick together. Please write this down. Please just take a moment to pause before you take off. The other strand, and I'm not gonna put a double helix here. It's a 3'. The other has to be anti-parallel. It has to be anti means opposite. So it has to be opposite the 3 on that side and the 5. So where you see a 5 here, the opposite must be 3. They have to be parallel, which means next to each other, and they have to be anti, which means the 5 and 3 have to be the opposite sides. Yeah.
Aren't the strings like coiled to the...
They are. I'm not doing it right now because I want to show you the hydrogen bonding. I'll coil it in just a second. So it's a good point. So A is going to match with T. I'm doing DNA here, not RNA. And wherever you see an and there's going to be a C here and a G here, etc., all the way down. And for every A and T, there's going to be 2 hydrogen bonds. And for every G and C, there's going to be 3 hydrogen bonds. All right. Where are we going? Sit down. No, you sit down.
We literally only get 20 minutes.
I know. But she spent an entire period on her phone. And then, but then gets up before I'm done. Sit down. No one else is getting up. Just sit down. You're wasting time. Sit down. So the 2 molecules wrap around each other the way someone just mentioned in what we call a double helix. That's it. That's all I wanted to say. I wouldn't have been so rudely interrupted. All right, have a good day. There might be some apples left. I don't know. Somebody said there wasn't, but I have protein shakes if you want them.
There's 5 left.
There's 5 apples left. Here's some protein. I love these protein shakes. What do you think?
You got protein shake?
I had surgery. I have a problem absorbing. I also have trail mix. Do you want some trail mix with banana chips and nuts?
Sure.
And then there's these things. And I have problems absorbing protein, so could you put these over there so people can use them if they want?
Yeah.
Yeah.
For the chromatography lab notes, did you want us to
I was taking it for your book.
Of them, not me. Don't put me in
I
this.
wasn't. I don't take it. Are we
All right, but just don't take me from that, thank you. Huh?
We gonna finish the conclusion on this or no?
No, I just want you to write your observations and upload them. That's all I want you to do with the chromatography lab.
That's all.
That's it. I don't need a lab write-up for that. I just want to see what your observations are. What did you observe? What did you didn't observe?
Okay.
There he is, the man of the hour.
Turn the lights on, will you?
Hey, Miss Elsa.
Hey, how you doing, Darryl?
Is it good?